Today, I wanted to discuss an exciting new story we have out on using gene delivery of immunomodulatory cytokines to the lung to preserve respiratory function, which has just been published in Science Immunology. We started this project back in 2020 during the COVID lockdown. We had a global pandemic focusing our attention on respiratory inflammation and a promising new PhD student, Tombi Makuyana, joining the lab. It was essential personnel only, which didn't include me. So James and Tombi teamed up to develop a novel anti-inflammatory approach. At this point, we'd only just moved over to Cambridge, and I still had a team working in Belgium. The team there were processing clinical samples from that first wave of COVID patients, so we already had a very good idea that the respiratory failure was driven by excessive inflammation from the immune system rather than the response of the virus itself. We had been working on regulatory T cells, or Tregs, and knew that they were a polypharmacy of anti-inflammatory agents. Our work demonstrated that there were low numbers of Tregs in pretty much any organ we looked at, and we thought if we could boost their numbers, we could drive local immune control and also aid the repair process in organs such as the lung. Fortunately, from our earlier work, we had a good idea of what was limiting the number of Tregs in the tissues, the amount of IL-2, or interleukin-2. IL-2 is essentially Treg food, and they need a constant source in order to survive. At this point, we had unpublished data that we could successfully boost Treg numbers in a tissue by using a viral vector to increase local IL-2 production. It was working well in the brain, so we looked to develop an analogous system in the lung. It took quite some troubleshooting, but Tombi and James developed a viral vector system that worked well in the lung. It is an intranasal delivery of AAV 6.2 coupled with the club cell promoter, and it gives lung-specific expression without any detectable production in off-target organs, including the draining lymph nodes, which will become important later on. By this point, we had new therapeutics and a vaccine approved for COVID, so we never actually tested our approach in SARS-CoV-2 infection. But COVID isn't the only respiratory infection that kills via inflammation. So we turned to one of the major unmet needs of respiratory medicine, influenza-associated pulmonary aspergillosis, or IAPA. While a very minor infection in healthy individuals, during co-infection with influenza, aspergillus can be a major killer, with around a 50% fatality rate in ICU patients with both influenza and aspergillus infection. So we teamed up with the KU Leuven group in Belgium to test our lung Tregs in IAPA. We used a mouse model of influenza-associated pulmonary aspergillosis that gives a very low dose of influenza infection, followed a few days later by aspergillus infection. The combination leads to severe health issues. We can measure this using measures such as loss of weight, and we found that the gene delivery of IL-2 to the lung rescued 50% of the added body weight loss caused by the dual infection. Likewise with the cumulative health score. Mice got sick with the flu infection alone, but the combination makes them very sick indeed. Giving the lung Treg boost to the co-infected mice returned them back down to the regular flu infection level. A similar story presents with lung damage. The lung is moderately damaged by the influenza infection alone, severely damaged by the co-infection, and the lung cytokine delivery rescued most of the extra damage during that co-infection. Finally, we also see a partial protection from inflammatory neutrophilia, one of the key drivers of respiratory pathology in this model. Perhaps most importantly, we were able to rescue the immunopathology in each case without releasing pathogen control. This is most likely because we were restricting the anti-inflammatory cytokines to the lung tissue, leaving immunity generation intact in the draining lymph nodes. So antiviral T cells and B cells could still be efficiently primed. This win-win scenario is why tissue immunology is such a promising strategy for therapeutic development. We can pick and choose which immune responses to shut down and which to preserve, based on which are harmful and which are helpful. I want to thank everyone in our amazing team and all of our great collaborators for working towards this milestone paper. In particular, Tomby and Laura, who led the research, and Gretchen and James, who guided the study with me. Finally, a big thanks to the ERC and Wellcome for supporting this work.